Welcome to part 1 of our video series on ICP OES. ICP OES is an acronym that stands for Inductively Coupled Plasma Optical Emission Spectroscopy. In part 1 of this series, we will be focusing on just some of the main principal components of this instrument. We will also look at the sample introduction system, and we'll talk a little bit about some of the safety considerations that we should be thinking about when using an ICP. Right now you are looking at an image of the ICP setup that we have in the Chemical Technology Laboratory. As you can see, there are several different pieces of equipment. There is a computer, there's the actual spectrometer itself, we have a cooling station down here, we have a gas tank, and we also have a waste container. So let's look at each one of these components individually and try to make some sense of them. The spectrometer itself is about the size of half of a bench top. This is a very large piece of equipment, and as you can see, there is a chimney and then a fume hood over top of it. The purpose of this fume hood is to ensure that none of the metal vapors that are produced during the analysis make their way into your breathing atmosphere. They should all be removed. On the floor next to the spectrometer, you'll find our cooling system. This particular piece of equipment is capable of cooling down a fluid and passing it through the spectrometer to cool the spectrometer. We will be talking in the second part of this series about why it is that we need this cooling system and how the cooling system is routed through the instrument. The gas tank that's standing next to the instrument looks a little bit different from the gas tanks that you're used to seeing. This particular tank is much taller, much greater in diameter, and much heavier. Please be careful when using these types of gas cylinders, which are called dewars, because they do pose a huge risk if they were to fall over on top of you. This particular dewar contains liquid argon, and we harvest the gaseous argon that's produced as the liquid argon is being evaporated. As we zoom in on the top of that tank, you'll notice that there's one other big difference between a dewar and a regular gas tank. Notice that there are multiple ports. Okay, so you will see there's a port for the gaseous argon. There's a pressure release port here. In the back there's a liquid argon port, and this is actually a pressure setting port right here. So depending upon what your applications are, you want to make sure that you use the correct port. Next to the argon tank, you'll find our compressed air system. As you can see, this is the same compressed air system that we use with our AA, our atomic absorption spectrometer. This particular system is set up so that the compressed air line is split between the AA and the ICP. It's important to realize this because the AA and the ICP cannot be run simultaneously for this reason. Also, the ventilation systems for these two instruments are set up on the same line. So again, for ventilation purposes and because of the compressed air, we cannot run both the AA and the ICP at the same time. Here's a view of the ICP spectrometer from the right side. There are several different components on the right side of this instrument. The first one I want to call your attention to is the on-off switch. Right now the switch is in the off position. If it were in the on position, there would be a red color part of the switch showing, indicating that the instrument is on. Please note that you have to push this switch very hard to make sure it flips over to the other side. Next to the on-off switch, you'll find one part of the interface between the spectrometer and the cooling system. Basically, this is just sending a signal to the cooling system telling it to start up. Next to that, you will find the ICP cord. This is what's going to allow your spectrometer to communicate with the computer that is attached. And up here is a service port. We'll talk a little bit more about this later on when we talk about maintenance in the second part of this video series. But right now what we want to do is focus in on this small panel on the back of, on the side of this instrument. You'll notice that there are 5 lines that go into the instrument. On the bottom there are 2 larger hoses. These 2 hoses are attached to the refrigeration system. So in one hose we have refrigerant flowing in, and in the other hose we have refrigerant flowing out. You'll see that there is a port where argon can be attached. There's a port where nitrogen can be attached. However, we do not use nitrogen in our system. So actually argon is being split and added to both of those. And finally there's a line where we can put our compressed gas, our compressed air I should say. We will be using this as a shear gas, which we will discuss further as we get into the second part of the series. On the other side of the instrument, you'll see our sample introduction system. Here's a close-up of how the sample is introduced into the instrument. Note that there is a sipper straw much like the one that you see in the atomic absorption spectrometer. Just like in the AA, the sipper straw is put into your sample and sample is drawn up. One big difference, however, is that the ICP does not use a vacuum to pull up the, well, it does pull a vacuum, but not due to the same principles that the AA uses. Instead of using the Bernoulli principle, this instrument is making use of a peristaltic pump. You'll also notice that there is a waste line, and that leads down to a waste container. This waste line is a little bit different from the one that we see for atomic absorption spectroscopy, because in this case we do not have any interlocks that are connected to the waste container. You'll also notice that there is a yellow hose that is leading inside of the instrument. This is where the sample is being carried up into the sample or into the instrument. And there's a clear hose coming out, and that's the hose that is going to be transporting the waste back out into the waste container. Now, as we look at this peristaltic pump, what we'll see is that there are metal rollers, and these metal rollers are going to spin around. Actually, it spins in a clockwise rotation, and as it does that, it's going to draw the sample up and push it up into the instrument. And then all the leftover waste is going to come out here, wrap around the peristaltic pump, and then go down to the waste container. Now, when we open the door right above the peristaltic pump, we can see the spray chamber for the ICP. You'll notice that there is a hose leading into the spray chamber coming from the sample. There's also another hose labeled nebulizer. This is some argon gas that's being pumped into the spray chamber, which assists in the atomization process. You'll also notice that at the bottom of the spray chamber there's a hose coming out, and that's the hose that's going to lead to our waste container. Here's a close-up of the spray chamber, also known as a Scott spray chamber. Please notice the exclamation mark right here. This little piece of plastic that is holding the spray chamber in place should not be removed while the instrument is in operation. When the instrument has been safely turned off, you want to lift that latch and push a small button that can be found at the bottom of this this piece right here. And as you do that, you should be able to pull the spray chamber straight out Make sure you do so carefully, not to snap off the sprayer right here. And here's what the spray chamber looks like when it's been removed from the system. Again, you'll notice that this is where the sample is going to be traveling through to be introduced into the plasma. This clear line right here, this is the nebulizer gas, or the argon, coming in. And this yellow line is our sample, which is being pumped in. By removing the knurled nuts holding the top of the spray chamber on, we can remove the top. And again, you'll see here, this is the top part. This is where the screws were. There's another screw on the other side here, or there was. And you can see coming straight in here, we have the nebulizer gas, which is argon, and our sample, which gets pumped in this direction. Notice that they are orthogonal to one another. They are at 90 degrees to one another. You can also say they're perpendicular to one another. Now, when we look inside the cap of the spray chamber, you'll notice that there are two nozzles. You'll remember from the previous slide that the argon was being pumped in through this nozzle, and our sample was being pumped in through this nozzle. So you can imagine that as the sample is being pushed in here, it's going to come out this nozzle, and it's going to meet up with a very strong stream of the nebulizer gas, which is argon. It's important that you check to make sure that these two sprayers are not clogged. It's very difficult to determine whether or not they are clogged, because sometimes if they have a partial clog, they'll still work, but you'll wind up having errors in your data. Now, if we look at the bottom part of the spray chamber, you'll notice that it has something that kind of looks like the baffles that you would see on the spray chambers of an atomic absorption spectrometer. Thank you for watching this video on ICP OES. Please be sure to practice looking at all the different components of the ICP OES in our laboratory. Make sure that you are familiar with them. Also, make sure that you are aware of the safety concerns that are associated with an ICP. Thank you.